Hey, what's up everybody, and welcome back to the Sanctuary. I'm Professor C, and today we'll be doing more anatomy and physiology. Specifically, we're going to look at the fourth and final group of organic molecules, the nucleic acids. So, without any further business, let's get to it. Alright, the nucleic acids, the final group of organic molecules. We already did carbs and lipids and proteins. Now it's time to finish it up with the nucleic acids. My favorite group to talk about, actually. Na, if you've forgotten, stands for nucleic acid. And then the D and the R in front of it are going to tell us something. But just to spell it out, deoxy ribonucleic acid and plain old ribonucleic acid. So we're going to talk about structure and function of these very, very large and very, very important molecules. Okay, we can start with the broad concept called the central dogma. And I know I don't usually tell, funny stories in here, but I have to share one with you. I had a test question once on a big exam, and it read something like, briefly explain the central dogma in your own words. If you're having trouble, draw a picture to help illustrate your thinking, or something like that, right? So one guy's exam had a half dog, half man, remember? A dogma, right? Half dog, half man, standing up and yowling at the full moon. So to give you a little bit more colorful context there, the man was the front half and the dog was the back half, and it was standing on its hind legs, its dog legs, while its human torso and head stood up with its arms stretched out toward the full moon. Its head was thrown back and its long hair was falling down its back. Its mouth was open and it was howling at this big, bright, glowing moon, and that was his dogma. I gave him half credit because it was the funniest thing I had seen all week. But that is not actually, I'm sad to say, what a dogma is. It is not a half dog, half man. It is a belief system or a tenet, an idea that happens to be so important in biology that it's called the central dogma of biology. Now, the details are something we're going to say for other courses, but I will give you the general concept here because it's very good to know. It begins with DNA. DNA is usually kept in the nucleus of a cell where it's safe and protected, and on it is the instructions to create proteins. But we can't simply go from DNA to a protein. That's not the way that the system is encoded. Our first step involves something called transcription, which will occur in the nucleus of our cells where we find the DNA. What we're going to do during transcription, again, we're not going to do the details, is DNA will be copied from a double-stranded helix, a double helix, into a single-stranded molecule called RNA. And then this RNA will leave the nucleus and enter into the cytoplasm, where it will be involved in what are called translation events. This is where we will read, the RNA will read this thing called the genetic code that you've probably heard about, and we will decide how to build a protein from that. We will, as a cell, decide which amino acids to put in the correct sequence, link them up by peptide bonds, fold them up into these structures, and then we have a functional protein. So the central dogma of biology has to do with the information pathway from DNA being the information molecule to protein being the final product. But there is the middleman called RNA that plays a huge role in this process. We can't just jump from one into the other. We have to do it in steps. All right, if you want to learn about any kind of polymer, which we know these organic molecules form polymers quite readily, you need to know the monomer. Okay, the monomer of a nucleic acid, whether it's DNA or RNA, is called a nucleotide. If it was DNA, sometimes it's called a deoxyribonucleic nucleotide. Sometimes if it's part of an RNA polymer, it'll be called a ribonucleotide. But the point is the building block of DNA or RNA is a nucleotide. Now, we don't usually draw a nucleotide like a ball and chain, ball and chain, ball and chain like we got away with. We could do that with carbs. We could do that with proteins. We could even do that with lipids to some degree with our caterpillar drawings. But to say that this little chain that I've drawn there in red is DNA is far from the story. A nucleotide, if you look over at this part of the slide, is made of 3 components. It's got something called a phosphate group, which we've seen before. This little circle P guy that keeps popping up on a bunch of different slides. Remember, it's just a PO4 functional group, but we just draw it as circle P and we call it phosphate. That'll be the least interesting part of the story because it's just going to form the backbone. You'll see in a moment. So phosphate group, check. One part of a nucleotide. A sugar, and that's all it says in the picture here is sugar, but very clearly it must be a pentose sugar, and you see the nice pentagon drawn there implying that it's a pentose sugar. It could be a deoxyribose if it's a DNA nucleotide, or it could just be plain old ribose if it's an RNA nucleotide. Here we're just going to call it sugar to keep it safe. Now, it also, a nucleotide would not only have a phosphate group, a pentose sugar, but it would have the third component, some sort of what we call a base, which contains nitrogen, a nitrogenous base. And it gets a little more complicated here because there are 4 different choices and 5 if you want to get picky. So we just need at this point to think of the monomer of DNA and RNA as being a nucleotide, and is made of 3 parts: a phosphate group, a pentose sugar, and a nitrogenous base. Let's draw a DNA molecule out, at least a little piece of one. A real DNA molecule could have millions and millions and perhaps billions and billions of nucleotides in it. And honestly, we don't have enough minutes in our life to draw that. So let's just draw a very simple picture of a DNA. Okay, I know that it's made of nucleotides, but I can't just draw the nucleotide as a ball now. So I need an actual 3-part nucleotide. So I remember it's made of a phosphate group, which I can draw as a circle P. I know it's made of some sort of pentose sugar. That would be a sugar with 5 carbons. And since it's DNA, I know the D sugar is what's going to be here. Deoxyribose will be the sugar because it's called DNA. And then, of course, we need the base. The base is the 3rd part, and it will always hang off of the sugar at a 90-degree angle. It won't hang off the phosphate ever. It'll look something like this. Now, I could just put a B in there for base. I could do that. But if I want to actually complete my picture of DNA, we can kind of cheat ahead a little bit, and I can tell you the bases of DNA are G, A, T, and C. And we'll define those better in a moment, but let's just say G, A, T, C are the 4 possible choices of a nitrogenous base for DNA. Let's just put a G here. It will stand for guanine, we'll see. And that's all well and good. We have a single monomer now. We have 1 nucleotide drawn on the picture. That's of course not a polymer, so that's not RNA, that's not DNA, that's not anything at this point. So we need more monomers, we need more nucleotides if we're going to make a polymer of deoxyribonucleotides. So we need to hook up another nucleotide, another phosphate group, another pentose sugar. In this case I know which one it is. It's deoxyribose because it's DNA. And then I have a base sticking up, hanging off of the sugar at a 90-degree angle. Well, let's just pick the next base it could be. I know it could be a G, but it could be an A for adenine. We'll see on the next slide. So let's add the base like that. Now I've got 2 nucleotides. Again, not an entire DNA molecule, but we don't have time to draw ones. But we're on our way. We're polymerizing monomers. We're taking monomers and turning them into chains now. Well, let's keep going. Let's add a third nucleotide. Another P, phosphate group, and another pentose sugar, deoxyribose. That gives us place to stick our third base here, a C for cytosine. Okay, and then we can repeat it to literally, sorry for this, to cover all our bases. I know, it's a terrible pun. There we go. Another phosphate and another pentose sugar called deoxyribose. Here we can stick another base, our fourth base, and the one we're missing is T, so we can put the T right here for thymine. GATC, the order doesn't matter. We wanted one of every base represented, so like I said, we can cover all the bases as we start to draw this. And it would continue. I could just put, dot, 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 and you could keep on going for millions and millions and millions if you had nothing else to do. But this is only half of the story. If you look here, what we call a backbone of the DNA, or sometimes better, it's called a strand, where you have sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. Sometimes you hear it called a sugar phosphate backbone, but this is only one single strand of DNA. If you really want to make DNA and talk about a double helix, you have to have two of these strands. And that's why I've left room on the top of the slide so I could draw the other half of the missing DNA molecule. So let's do it. In order to make the other strand, we have to kind of make a mirror image as best we can. But I know the easy part is drawing the backbone. The backbone is simple, just phosphate, sugar, phosphate, sugar. So let's do that first. Let's just sketch out a backbone. Okay, bling, there it is. A sugar, phosphate, another backbone, another strand. So now we've got a double-stranded molecule. We just need to attach one to the other somehow. So we can attach them, but we're kind of missing something very important. We're missing the bases up top. And I know the bases are going to hang off here, off of the sugars, never off of the phosphates. This one's kind of off kilter, but we can deal with that. There's room for 4 bases, but which ones go where? Well, there's a guy named Chargaff, and there's some rules named after him. And I'm just going to tell you the rules so we don't have to go over the history of it. But what Chargaff said was, wherever there's a G on one strand, right across from it, there will always be a C. Guanine and cytosine always pair up if they have the chance. So I'm going to put a C here, because I know it will hook up with that guanine. And not only that, he said that the G and C pair, what's called a base pair, are held together by 3 hydrogen bonds. And we can draw a hydrogen bond by simply making a dotted or dashed line connecting those two objects. And there's our famous hydrogen bond. A very weak bond, but there's 3 of them there in that base pair. Well, next to it, which base is going to go here? Well, there's an A down on the first strand. Chargaff said that, C goes with G, so that must mean that A will pair up with T, and it does. So a thymine would go in here. And they're not as strongly attracted as the CG pair. This base pair, Chargaff found out, was only separated by 2 hydrogen bonds. And now we can complete our DNA molecule with no problem. We can put a G across from the C, because I remember the rule. There's going to be a triple hydrogen bond holding those 2 together. And then the final box will be a what? It'll be an A, right? Because it's across from the T. And there will be how many bonds? 2. And now you have a really, really nice picture of DNA. How many nucleotides are in this drawing? Well, there are 8 of them. And I'll draw one in a circle real quick. There's a nucleotide, right? Basically, everywhere you see a base, you can assume the rest of the nucleotide is there. So if you know how many bases are there, you know how many nucleotides are present. Now you could keep on going, thousands and thousands and millions and millions and millions of these, and create, an average size DNA molecule, but we don't have time for that, right? Imagine this image in your mind as I erase it. And then imagine kind of getting farther and farther away from that picture, where it starts to kind of look like 2 sides of a ladder with the rungs of a ladder joining them together. And this would be what we call one of the strands. And there's another one here. We got to have two. And then the base pairs would be here. C with G and A with T. And then if you could take that latter image and you could twist it in space, you'd find out that one of the sides of the ladder is twisted around the other side of the ladder, and the rungs kind of start looking like this. And then you get that famous double helix picture that you see in so many of the images on TV and the internet. Okay, so here's another view of DNA, kind of with a whole bunch of little factoids here that I mentioned probably just now, but let's make sure we cover all of them, all the bases again. So DNA, I put the 4 bases up top here: guanine, adenine, thymine, cytosine. I underline that T because it's going to be unique to DNA. When we get to RNA in a moment, we're going to find out there's a different base there. There's a substitution there in RNA. But if you see the T, it's a giveaway that you're looking at DNA. So DNA is found in the nucleus. We went over that. We said this nucleus keeps this information safe. It is double-stranded. It is helical. And if you drew that picture along with me, you should be able to see in this image the sugar phosphate backbone. No problem. And the 2 bases, T with A, double hydrogen bond, C with G, triple hydrogen bond, and so on and so on. It's the same picture we just drew, just slightly different. Because this molecule is so big, we do need some packagers to help us move it. Proteins called histone proteins. What is the function of DNA? Well, let me just sum it up with what I've said a few times already, I feel, during my talk here. It's information. It's information. DNA is information. Information for what? For building proteins. And we saw the last talk when we talked about how important proteins were and how many major classes of protein function there is. A cell can't work without its protein set. DNA is the instruction manual to create proteins. Later on in other talks, when we get more complicated, I'll talk about it being a recipe book from which we can cook up some proteins from the information inside. It will replicate itself. We'll talk about that in the 3rd unit in here when we talk about cells and cell cycles. And of course the bases, don't forget, GATC are the DNA bases. Okay, we got some RNA here in this picture. It kind of looks just like a comb. Kind of looks like half of a DNA, and that's exactly what it is. If I can change my color here, I could draw like a ribbon, like there's one of my strands, and the bases just stick out here like teeth on a comb. And so if you see this ribbon looking thing, it's RNA, a half of a DNA if you want to go there. There are some differences more than that. It's not just half of a DNA. Notice here, there is no T. There is no thymine. So instead of GATC, like we saw in DNA, in RNA we'll have GAU. Uracil will replace thymine. They're very close in structure, but not the same. So it is a single-stranded molecule, a single-stranded nucleic acid. It is mostly active outside. Remember, we talked the central dogma that DNA is copied into this RNA form during transcription, and then the RNA leaves the nucleus and works in the cytoplasm to actively create the proteins. So 4 bases GAUC, uracil, instead of thymine. There are a whole army of these guys if you want to think about it. I think about cooks inside of a kitchen, cooking stuff up. mRNAs, tRNAs, and rRNAs. We're not going to get into the specifics in this talk, how they work. We'll have to wait for a later day, unfortunately. But there are multiple types of workers called RNA that carry out, it says there, the DNA's orders for protein synthesis. A good little comparison showing the 2 molecules side by side. It shows not only single-stranded RNA versus double-stranded DNA. It shows GAUC are the bases of RNA and GATC are the bases of DNA. So nice picture showing the details. But most of the questions on standardized tests are not pictures. They're some sort of factual information. So I made a little chart down here that kind of helps us straighten it out. What's the difference between DNA and RNA as far as number of strands? Well, DNA has 2 and RNA has 1. What is the difference in the sugar that makes up the backbone of these molecules? They both have a pentose sugar, but DNA has deoxyribose, the D in DNA, and RNA has simply ribose, the R in RNA. Bases in DNA, GATC. Bases in RNA, GAUC, uracil instead of thymine. Okay, the point of this slide, and again, we've seen this ATP monster in several different places, and the whole story never will come out until we get through many, many, many chapters in here. So let's tell the simple story of ATP. So look closely at this structure. I see a circle P group. There's a phosphate. I see a pentose sugar. And I see a nitrogenous base. This one happens to be adenine, base A. So not only do I see these three things, they're all hooked together. And if I see a phosphate group, a pentose sugar, and a nitrogenous base together, this is what we call a nucleotide, the building block of a nucleic acid. The only thing here is this molecule that I'm looking at will not form chains. It will not form polymers like we saw DNA and RNA do. In fact, this thing would be called AMP, adenosine, and you can see that word right here I'm underlining. And instead of triphosphate, that would be called monophosphate, AMP, because it only has one phosphate attached to it. It's a simple nucleotide, AMP. However, if you were to add a second phosphate group here, instead of just chaining nucleotides, just literally add another phosphate on the end of it, now it becomes a DP, right? Adenosine diphosphate. And this second one that's been attached makes a very high energy bond, and we can store a lot of energy there. Remember what bonds are, is a place to store energy. We can store it there, save it for when we need it, and rip it apart later and retrieve it. But we can go farther. We can add a third phosphate group with an even higher energy bond that we can store a lot more energy in and save it for a rainy day. This would be adenosine triphosphate that has all three of them on it. Again, the purpose of this is not to understand ATP just yet, but we're getting little samples of it as we go through here. The point mainly is to show you that ATP, ADP, AMP, adenosine triphosphate, adenosine diphosphate, adenosine monophosphate, these are all nucleotides. And it says what we can do. We can store energy in those phosphate bonds and perform a lot of cellular work later. This all has to do with phosphorylation, and we'll do the big, ugly, scary story much later. Okay, as usual, thanks for watching the video. If you liked it, check out some more in the series, and I will see you for the next one. Bye-bye.